All right, guys, let's go ahead and get started today. Wow, that's so loud. Okay. All right. Oh, much better. Okay. We're going to start. We're going to be talking about what we're doing next week, which is our actual unknown identifications, utilizing the skills and the tests and stuff that we've used before. So, more or less, it should all be stuff that you're familiar with. It's not a shocker. It's nothing like crazy that you haven't done before. So let's talk a little bit about this. Okay, so first things first. You're going to have either a gram positive, you're going to have a gram positive and a gram negative. For your gram positive, you need to do a catalase test for it. So for your unknown tube, you're going to do a catalase test on it. You're going to determine if it's catalase positive or negative. If you have a catalase positive, you'll go up to your TA and double check that it is correct before you go on and grab the media for these guys, which is a Mueller-Hinton plate, a DNase plate, and an MSA plate, as well as a Pyre broth. If, however, you go up and say, hey, look, it's catalase negative, you're going to grab a BEA slant, a Pyre broth, and a blood auger plate to do SXT antibiotic sensitivity test on. Those are the two different types of tests for gram positives. You have to check with the TA before you grab any media. If you grab media that's not yours, that presents a problem because now somebody else doesn't have their media and then they can't do anything. So please be careful not to grab the wrong kind of media. Check with your TA about your catalase test before you do anything else. If you don't, then you're going to run into a big problem. Next one is gram negative. For your gram negative, you guys will get a suspension, inoculation broth. You will take a sample from your gram-negative tube, put it in there, and swirl it around so it's evenly distributed, and then you guys will inoculate it. For you guys will be using the Rapid 1 ID tests. Basically, you're going to follow the same procedure that we followed this week. Pretty easy. Stick it in the incubator, let it hang out for a bit, and then we'll read the results the next day we have class. Now, with that said, most important things to do here to get the best results, to get the identifications correctly, is make sure you're very careful to use aseptic technique, because if you don't, you're going to run into a lot of contamination issues where it can basically alter the results incredibly, like just a ton. Perfect example of this is catalase test. If you guys remember catalase, it's an enzyme that most living things have, but a couple don't. And those couple that don't, they're very peculiar, and so they're easy to identify based off of that. They're mostly Streptococcus species. The most important aspect of that, though, the catalase negative ones, catalase is such a reactive enzyme that even the catalase that's on the bacteria or just being produced exogenously on your skin will alter the results. You can get a catalase positive test with a very minimal amount of contamination because the enzyme is so active, fastest enzyme known to man, so be careful with that. Okay, so we've got our gram-positive tests, new and improved. These are a little bit more clear. Hopefully this will help you guys do identifications. We're going to talk about the important things to look at for identifications, the ones that are the most important to go, hey, this is what this is what it isn't. Because I know that there was a little bit of difficulty trying to identify these last time. That's okay. We're going to help you guys out with these. First one that's really important for the catalase positive tests is our mannitol fermentation. So if you're going on there, that's great and all, but if you're able to ferment mannitol, that really helps differentiate you. So for example, if you are a non-mannitol fermenter, these are your only three options. If you're a mannitol fermenter, these are your only three options. Pretty simple. The ones, and Pyre broth is a little hard to read. Sometimes people get it wrong, that's okay. This is the really important one that's actually really easy to read because that yellow zone's super bright, super easy to see. And of course, growth is obviously very easy to see. Now, some other things that are very, very helpful, are growth characteristics. But most important fact from here, growth characteristics can't be read on dyed media. And that's actually really important so that you guys know that. I think that there was an issue with people going, oh, I'll just read the color of the bacteria off the MSA plate. But if you're MSA positive and mannitol positive, you're going to be yellow in color because the dye changes the color of the bacteria. You can only read the color of the bacteria off of a clear plate like TSA slant or a mannitol agar plate, or DNase even. Those are all clear, non-dyed plates or media so that you don't alter the color of the bacteria. So that's really important. Within these, there's some easy ones. So if you had something that's kind of got this light yellow and it's susceptible to novobiocin, that's going to be Staph aureus. But it's a very light yellow. It says yellow. It's kind of this light gold color. Staph epi, very similar in structure to this, but it's white. These two are really easy to differentiate because they are both very pigmented and they look like tetrads when you guys gram stain them. So if you looked at your gram stain, you're like, hey, there's this repeating pattern of 4 everywhere. Boy, it's probably one of these two. So Micrococcus luteus, it produces a bright yellow coloration. Micrococcus roseus produces a light pink coloration. So if you see pink bacteria, and on the microscope they are in a tetrad formation, they repeatedly produce that pattern of 4s, then you're gonna be able to look, oh, that's that one. It's really easy to identify. Okay. Staphylococcus psilocysis and Staphylococcus saprophyticus. The most obnoxious out of the two, I know. The real only major difference that we can say is, with the updated chart, is that one's going to be pyre positive and one's going to be pyre negative. And it's just kind of an obnoxious one. Be very careful with your pyre test. Don't add too much liquid, either the reagent or the broth, so just be careful with that one. Okay, now the next one is the catalase negative tests. The most consistent one is esculin. It's really hard to have an error on the esculin test. So there is one variation with this one that I want to talk about. So within the esculin test, so you've got strep zooepidemicus, streptococcus, galeoctus, and xylosis. And these ones are great, but sometimes it'll be kind of this light, faint coloration. It's not going to be as strong, especially with Enterococcus faecalis. Especially with zooepidemicus. That one tends to be just a slight weak production. So if you got one that's kind of like, yeah, it's growing there, it's creating that kind of dark coloration, it's going to be kind of light, that's okay. That's normal. Okay. Now the other one that you do want to be careful with but does give you the best results is these alpha, beta, gamma tests, okay? If you're beta, and this is going to be a strong beta change, it's going to be Staphylococcus pyogenes. If you are, for zooepidemicus, sometimes it can be beta, and this is kind of the unfortunate consequence of having incubation times that are like 48 hours. If it sits there really long, it'll sometimes become beta, so just be aware of that. But mostly it sits as alpha. Streptococcus mutans, easiest one, it's none. It doesn't do anything. It just sits there and chills and doesn't change anything. Enterococcus, Galeoctus, Agalacti, and Faecalis all do alpha, but you can see some differences, especially with the pyre broth on those ones. Okay. Does anyone have any questions on the gram positives? So those are the key tests. Yes? Really, the only one that does that typically is going to be zooepidemicus. If you basically let all of these just sit on a plate for like a week, the ones that are alpha will eventually become and look like beta because they've had long enough for the enzymes to break up all the cells. So there's kind of this like range of, hey, this is when I should read this. That's really the only one that's going to be tricky for you guys for this sense. But basically, if you see something that's, esculin positive, it can't be pyogenes. So you just chuck that one out, okay? All right, good question. Okay, gram negatives. We also have new improved gram negative identifications. So I know that the REML one wasn't working out as well for you guys. That's okay. It's our first run through with it, and not everything is always going to work out nicely with it. I know this chart is really hard to read. This chart will be available for you guys to see on your lab archives, but we're going to do a little zoomed in version here. So these are the options of bacteria that you have for this test. And it kind of covers a pretty wide range of them, but we're going to talk about the most important aspect, which is TSI, which is the most consistent test, mostly because it's a really easy to control test for us and for you guys. It's like there's almost no way to mess it up, as long as you do the inoculation properly, which is the stab and streak method. So make sure you guys do that properly. The way it works, so I'm sure you guys noticed that some of them grew on there and they had fermentation. Some of them didn't have fermentation on the top of the slant, some of them only had it on the bottom, and some of them didn't ferment at all. This tells us a lot of information. First and foremost, if it doesn't ferment at all, that means it's going to be an oxidative organism. There are 3 oxidative organisms in here, which is Pseudomonas aeruginosa, Moraxella catarrhalis, and Alkylagenes faecalis. But that one does a little bit of weirdness. It sometimes alters a little bit of the media, just because when it's growing on there, it'll start dying and produce acid. So if it doesn't look like it's a strong like yellow, then you just go, that's just messed up. So I'll show you guys another way to differentiate that one. Those are the 3 oxidative organisms, so they're really easy to identify on the TSI. You're just like, oh, the whole thing's red. This guy has to be one of these 3 organisms. Easy test. The next easy test within here is H2S. There are only 2 H2S organisms that we will be using, or that we've used for our unknowns, which are Proteus mirabilis and Proteus vulgaris. But people also tend to confuse Pseudomonas aeruginosa. The reason why is because Pseudomonas aeruginosa will be red over red, so alkaline over alkaline. And because it produces a dye on it, it'll grow on the slant and it'll create this kind of black film on it, and people will go, oh, that's H2S positive. It is not. That is a trick that kind of messes people up. It's just the way the bacteria grows in there. For it to be H2S positive, it has to grow at the bottom of the tube and create a complete blocking out of all light. So it's a precipitate, so if you hold it up to light, you won't be able to see any light shine through that black precipitate. That means it's truly H2S positive. So keep that in mind. So if you do see one that's kind of got that red over red and it's got that kind of like black film on the top, and you're like, whoa, that's weird, that looks like H2S. Nope, that's probably Pseudomonas. Okay. These 3 tests, the ADH, ODC, and LDC tests, are not good ones to rely on, just because we've had some issues with them being consistent. We're not exactly sure why, so we're just going to let you guys be forewarned. Those 3 tests, not crucial to identifying them, identifying your bacteria, okay? Going down the list a little bit. Very important tests. TET and LIP, lipase. There is only a couple of organisms that are positive for these, okay? Pseudomonas aeruginosa is positive for lipase, but not for TET. Moraxella catarrhalis is positive for both. Morganella morgani is positive for TET, but negative for lipase. And Morganella also has an interesting ability. Morganella also has an interesting one because it is A/K, so it only ferments glucose and not lactose. So it's got a red slant and a yellow butt. And it also almost produces no CO2. You guys notice I didn't harp a lot on the CO2. CO2 is kind of a variable situation. Some bacteria produce a lot, some bacteria don't. The only ones that tend to produce a lot are like E. coli. So don't read too much into that. Okay. Let's see here. Next one up. So we got TET and lipase. KSF, the only one that tends to show up consistently positive is going to be Klebsiella. Oops. And I don't know why this one didn't highlight. Sorbitol is another one. So E. coli is going to be positive sorbitol very consistently. In fact, that's one of the key tests to identify E. coli organisms. There's only 2 others that are positive, which is Klebsiella and all the way down here it's Serratia. The next test is MAL. So if you're able to utilize this as your carbon source, that kind of narrows it down to 3 organisms, which are Klebsiella, Moraxella, and lastly Pseudomonas. So if you have something that's able to ferment from the TSI, but it's also positive for MAL, that's probably going to be Klebsiella because Moraxella and Pseudomonas are both oxidative organisms. If you're oxidative, you can't ferment. Okay, scooting down here. We just talked about the MAL one. The last little bit on here is cultural characteristics. Now this is a very important one because it gives you some information that's unique to each of these organisms that's kind of separate. It's not really a test so much, but it is kind of important. So I'll give you guys some information. The first one is Serratia marcescens. It's red. So if your bacteria produces a lot of red pigmented dye, it's probably Serratia because it's the only one that does that. If your bacteria produces a lot of green coloration on the agar, that's probably Pseudomonas because it's the only one that does that. If you have a COSI It is going to be Moraxella cataralis because it's the only one that is a cosi. Everything else is bacillus. Now, of note, one of them is called Alcaligenes faecalis. It has very, very small bacilli, and people often confuse, I don't have a drawing thing up here. Let's see. We don't have anything yet. We'll just have to describe it to you guys. The way it looks is it's going to be very small, and people often mistake it for cosi because it's so small. It's smaller than E. coli, so it's very tiny, but it's got kind of a squish shape. So it's not a perfect circle like a cosi is. It's kind of like this little teardrop shape. And so if it's not a perfect circle, it's not a cosi. If it's very, very small and it's not a cosi, it's probably alcaligenes. Okay, the last two that are kind of easy to identify based off of other characteristics are Proteus mirabilis and Proteus vulgaris. Proteus mirabilis has a high rate of spread on any agar it grows on, which is obnoxious. So if you've been growing it and you put it on some plates, TSA slant, and it just kind of swarmed the entire width of the slant instantaneously, and it kind of has these like what look kind of like waves. That is a type of motility that we have, and it is very unique to Proteus mirabilis. Proteus vulgaris also has this spread. It spreads out very far pretty quickly, so keep that in mind as well. And these two can kind of be differentiated from one another in a couple of different ways. The most important one is Proteus mirabilis can only ferment glucose, and Proteus vulgaris can ferment everything. It's A/A, so that's kind of its big differentiating things. Okay, does that make sense so far? I know it's a little bit complicated, but these are kind of these steps that you kind of have to go. Finding good tests to identify and then move on from them. Do you have a question? You didn't have a what? That's irrelevant. We're talking about our individual unknowns. Remember those slants that we gave you guys like a couple weeks ago? That's the ones that we're going to be doing next week. Okay. All right. Ignore all that. Okay, great. Okay. We're going to talk about cool science thing of the week, and then I'm going to let you guys go. So not a long day. You guys can get out of here early. We're going to talk about cool one. Well, okay, maybe not cool, but very interesting one. So uropathogenic E. coli, pretty neat stuff. It is one of the major causes of UTIs. And typically, I think UTI is something like 190 million people a year get UTIs. Some of them are actually quite dangerous. It can kill people. It also sometimes predicts the onset of things like, I believe, Alzheimer's. If you have a severe case of UTI, that can be a triggering event for Alzheimer's, which is unusual, but we've noticed that. So what is cool about this E. coli? Well, despite being a facultative anaerobe, as the authors state, oxygen plays a central role in its ability to colonize the bladder and form stress-tolerant biofilms. And although E. coli codes 3 separate oxidases capable of doing aerobic respiration, so 3 of them, not just 1, not just 2, 3, only one of them is important for this ability to infect bladders. If you lack that one, you cannot infect the bladder effectively, and it's kicked out of the system easily. It's really interesting stuff. So what do we have here? Well, we've got our picture right here. And in the purple is actin. These are bladder cells from mice. And then in the green, we have a bunch of E. coli. These E. coli are normal, nice, wild-type E. coli that are urinary tract infection type E. coli. These guys, though, have a mutation in that BD gene, the cytochrome BD, and if you notice, they look pretty different. The ones that are normal wild-type ones have these big clusters and chunks. These very thick biofilms are holding these bacteria together heavily within these cells, within the tissues of the bladder walls. But these guys, they are far more spread out. Even though there are similar amounts of bacteria, they are not clumped up. They are too spread out. They have not formed effective biofilms. Just by taking away one oxidase out of 3 that you have, it messes up the entire system. On top of this, what they found, the oxygen affinity of cytochrome BD is approximately 1,000-fold higher than that of BO and the human cytochrome C oxidase, suggesting that it promotes intracellular pathogenesis by allowing it to scavenge oxygens in the hypoxic cytosol. So what is it doing? It's stealing oxygen away from the mitochondria. It's scavenging all this oxygen around it to help it drive its replication and help it be effective. But it does one other thing as well. As such, we hypothesize that the intracellular bacterial oxygen consumption depletes cytosolic oxygen and stabilizes hypoxia-induced transcription factor, HIF1, which we'll talk more about in a second, and introduces a shift towards aerobic glycolysis. So they're saying, hey, we want to make sure that we set up our bacteria to drive towards aerobic environment, even though there's not a lot of ton of oxygen in bladders, but they do it for a very specific reason. So what ends up happening? Well, this picture's really, really neat. This is a normal mouse bladder tissue. The blue is staining, DAPI staining, so it's basically saying, hey, here's the neutrophil. The purple is for mitochondria, and the green is bacteria. And if you notice here, in this one, that's a control, so it doesn't have any bacteria, there's no bacteria. The mitochondria are evenly spread out. There's a nice kind of even coating of them, no problem. Okay. This guy with the wild type bacteria, the mitochondria is super depleted, like there is almost none there. I know this scale is really hard to read and fuzzy for some reason, I don't know why, but this is basically showing how big the mitochondrial spread is, and it starts at 20 here on the low end, it's down to 2. So it drops it heavily when you have these bacteria that are wild type in here, that basically depletes and destroys the available mitochondrial structures. When you have the ones that are BD negative, so they don't have that cytochrome, so you can still see the bacteria in here, we now have mitochondria that much look like the normal mitochondria. They're evenly spread out. There is some depletion, but it's significantly higher still. So it does alter things, but it doesn't alter them as much. Oops. Nope, that's not what I wanted to do. Oh, I guess I'm missing a slide. Okay. So why is it important? Well, remember we were talking a little bit about this guy right here, HIF1. What does he do? He controls bladder cell wall apoptosis. So if you mess up the mitochondria, you change the way they respond to this protein called HIF1. If there's not enough of it, then the cells don't slough off, because for bladders you have to constantly kind of replenish the bladder walls. And here's the thing, if you're a bacteria and you're trying to stick to a bladder wall to infect it, you don't want to constantly be sloughed off by dying cells. So they literally change not only the mitochondria, but also other signaling within the cell to make it stop sloughing off so that it can be better at infecting. It is actually kind of crazy to see how much they alter our own body's signaling to create a better environment for themselves. It's not just things that they do to resist our immune system, they're actually changing the way our body produces different types of protein and signaling to allow themselves to infect better. It's actually really, really cool. So this is kind of a new thing that we figured out, very recent. It was in Nature, I don't know, 2 months ago. So the article's there for you guys to read on Baylor's library website if you guys are curious, even though it's kind of boring. All you need to take away from this is, 1, aerobic respiration is really important for a well-known anaerobic, facultative anaerobic organism to create UTIs. And if you don't have the one cytochrome oxidase for E. coli, it reduces its ability to infect UTIs by a significant amount. That's all you have to take away from the school science thing a week. Okay, any questions? All right, I'll see you guys later. Have a good weekend. Oh, and no quiz this week because it's all stuff you guys know already, so you guys don't have to take a quiz. I assume you guys know.